It's my great pleasure to be here today with all of you celebrating these fantastic scholars, and also to introduce one of UC San Diego's scholars, Dr. Zhang. Dr. Zhang is a distinguished professor in the Department of Molecular Biology in the School of Biological Sciences, and in the Department of Pathology in the UC San Diego School of Medicine. She also has an appointment in the Moores Cancer Center, and her research studies the molecular basis of cancer and cellular basis of cancer, and how these interesting rearrangements and mutations that characterize cancer cells can drive the development of cancer. She is one of our most distinguished scholars, and she has been recognized by the Leukemia and Lymphoma Society, and she is also an elected fellow in the American Association for the Advancement of Science, which is a really fantastic award. And on top of those awards, she is also a fantastic instructor. She teaches the molecular basis of human disease, and she's also the chair of the Department of Molecular Biology. So please join me in welcoming Dr. Zhang.
Thank you, Dean Palmiano, for the introduction. And also it's my honor to come here to discuss about Dr. Axum Sunari's work, why the work is so impactful, and for him to get this really great prize. So let's see the next slide. So Dr. Sunari was born in Kenya in 1945 and received his early education in Kenya. Then he moved to United Kingdom and did his Doctor of Philosophy degree study with Sir Robert Edwards. Maybe some of you know who is Sir Edwards, and actually there is a movie made by Netflix last year called Joy, specifically talk about Edwards' work. He is a pioneer and in the human in vitro fertilization, and produced the first in vitro fertilization generated baby in 1978. That time period, there is a lot of challenge to do this, but he is a developmental physiologist, know a lot about early embryo development. And Dr. Sunari was his student in Cambridge, University of Cambridge, and during that time period, so he knows a lot about how to really control the fertilization process by using different kind of approach. Okay, so then with this really strong background in this field, his major contribution leading to today's award is the started the genomic imprinting, this concept, and also further developed the genes involved in this process and how this gene regulated by genomic imprinting. So, as we all know, for most organisms, the generation production is through the male and female, with most organism cells are really somatic cells, but there's still a little bit, a small number is germline cells. These germline cells basically is used to produce the offspring, and through a special cell division called meiosis to generate the one end cell. So that means instead of the diploid cell contains one gene from mother, one gene from father, and germ cell only contains one. Okay. Then this male one produce sperm, that's a germ cell, and female produce egg, germ cell, and these two fertilize, form the fertilized egg, we call it zygote. And this one through many cycle of cell proliferation and make the whole body. Okay, so, and then come to the genetics. What's really interesting about this is in Mendelian genetics, that's a classical genetic concept. The disease with gene mutation transferred from either mother or father. So when in this case, we're saying if they are like disease one, like people carrying disease will be solid covered, and with the bad gene but no disease will be with a dot. Otherwise, will be healthy, perfect gene. So there's two very famous diseases. One is Huntington disease, which is dominant inherited, and then sickle cell anemia is recessive inherited, which means one, as long as you have the gene, then you have a problem. The other one is you have to have two bad genes and you have a problem. But in this case, it doesn't matter the bad gene come from mother and father. For the dominant one, the person always got a problem. Okay, so then scientists are always very curious about everything. They try to really see what's really the possibility of not really this way. So they are organisms. They are not really depends on fertilization, can make a reproduction. Okay, in this case, the word called is parthenogenetic. So this means virgin births. So you probably heard about this. So they are algae, like insect, like a bee, or like a lizard. They can just produce without fertilization. The female egg can do the job. Okay, so but not really in mammal. We never really see this. Therefore, Dr. Sunari's group said, is that possible? Let we make experiment to really see whether it's possible. You can put change the nucleus inside the cell, fertilized egg. And he made a conclusion after all the trial. He said, no, in mammal, to use mouse, you have to have both male and female contribution, the DNA, to make this. Otherwise, it won't work. And how can that be? So he thought about something, he kind of the concept, some gene must be not really expressed. Either from mother not expressed or from father not expressed. You need both mother and father's gene, right? So then he called this genomic imprinting. So in 1984, he published this work in the top journal in the world. And take another 5 years, people realized what is the first imprinting gene identified. Okay, and this is very important. Why? Because this gave a field called epigenetics, which just means you can have the same DNA sequence in the gene, but the gene, the DNA could be modified, not by the DNA sequence, by something get on top of it, modified this way called methylation, DNA methylation in this case. So if the gene methylated, it can be genomic imprinted and not expressed. So then in this case, we can see, so the imprinted gene can be either is maternally imprinted, then only expressed paternally, or is paternally imprinted, not expressed in the maternal side. Therefore, if you only have the DNA from one side, it won't be developed because we're missing the express of the gene. So in this case, then there's another type of human disease, is the inheritance. Now the Mendelian style, this one is totally genomic imprinting related. In this particular category, this person kind of disease called Prader-Willi disease is a paternal expressed gene, maternally imprinted gene. Therefore, like in this case, the mother with the disease. But the gene come to the germ cell from mother's side were not expressed. Therefore, their children all fine, no problem, nobody really have disease. Okay, then come to the next generation. If her son got the disease, got the gene, not disease, then his children, if carried this by the gene, will have the disease. But their daughter with this by the gene, her children will be fine. So this is a totally different set of genetic inheritance pattern. So, and this really gave the scientists the possibility identify a lot of problems related to the genetic, not really valid inheritance style. In this case, very true, like the disease I'm teaching in my class is one is Prader-Willi syndrome, one is Angelman syndrome. So Prader-Willi syndrome is the gene, by the gene come from father. The Angelman syndrome, the by gene come from mother. So in Prader-Willi syndrome, so in this case, this is rare disease. The person after birth, one symptom is very reduced muscle tone. Another one is like increased eating, and one is after 1 year old, like young kid. But initially, because of muscle weakness, they cannot suck the milk. They have to really special feeding. So they generally die earlier. And if you look at them, you know, this is a newborn, not really strong muscle, and then during the early stage of life, they are overweight, have to really control their eating. And in Angelman syndrome, it's another rare disease, but in this case, its problem is a mental retardation. So the person really have a lot of issues, and most obvious problem is really the tremor, the speech, all this, and they suddenly laugh a lot. If we look at them, they always have smiling face in a way. Now means everybody look smiling face means they have Angelman disease, but these people, they do. Yeah. So then this really triggered the field to study epigenetics, DNA methylation, and other methylations like regular gene expression. And here shows after many, many years since the word genomic imprinting come out, the scientists identified the region in Axum, put 15 on both mother and father side. The blue one shows the father side should be expressed, mother side should be imprinted, not expressed. If those genes wrong, then it's disease from father side. From mother side, no problem. And then from mother side, you also can see the gene which is supposed to be expressed. If those wrong, is Angelman disease. And also, these genes, so far we identify about 200 genes called genomic imprinted gene. And they generally control the food taking, the growth, the metabolism, this type of situation. And also notice if their expression level have to be well controlled. Like this gene called UBE3A, if it's wrong, it's Angelman disease. But if it's too good, overexpressed, it could link to autism. So that's how the initial genomic imprinting really impacted the entire field and made a lot of new discoveries since 1984. So I'm going to stop here. Thank you for listening.